Okay, welcome everyone to this version of the Center for Evolution and Medicine Thursday seminar. It's a pleasure to welcome you and introduce today's speaker, Rob Dunn, who's with us from North Carolina State University. So Rob is a proud graduate of Kalamazoo College in Michigan, where he has a Bachelor of Arts degree. From there he went to UConn, where he studied with the terrific Rob Colwell, and then on to a Fulbright in Australia, Curtin University. He did a postdoc then at the University of Tennessee in Knoxville in an effort to hit all institutions in the free world. And in 2005 moved on to North Carolina State University, where he's in the Department of Applied Ecology. I had a delightful dinner with him last night talking about a whole range of topics. The kinds of things he's thinking about is just really terrific. His research, though, formally focuses on ecology and evolution of the daily life of societies, and it doesn't matter for him whether it's humans or ants, and he thinks about lots and lots of other stuff, including the man who touched his own heart. So it's not just a picture, it really is a book. He got the job done. He's published several books, and is working with Chicago on a whole bunch, University of Chicago on a whole bunch of others. So he's really a talented, thoughtful individual across a whole range of biological issues. And he's going to talk to us today about the lessons from the heart. Rob, it's all yours.
Thank you, Jim. I've had a delightful time here already. Thanks for having me. I left at 3:30 in the morning yesterday from my house, got here, talked to people, met with people, had dinner with Jim, and I'm still standing and happy, and that's a testament to all the cool things going on here. I'm going to give kind of a funny talk today, which is a talk about the value of ecology and evolution in discovery, and discovery as it ultimately relates to application. And it's a talk that emerges out of writing I was doing for this book. And it's funny that Jim says that it's an actual book, not just the cover, because I always get the covers before the book, and so there is this window when I can use them before they exist. But so this book ends up being, or I hope it is, sort of a subversive introduction to the value of natural history, ecology, and evolution, but where the reader doesn't know that's what they're learning about until the book sort of fills in toward the end. But I want to start off with how I got to the point of thinking about hearts and the value of these things that we hold dear, ecology, evolution, natural history collections, to application. And it really started for me when I wrote this book, Every Living Thing, which really kind of started when I was a graduate student. And it started in part when this guy, Peter Niskrensky, who was a graduate student with me, not at this age. This is Peter as a little boy. This is a lizard species that Peter accidentally introduced into Poland. Still lives there. But Peter was a grad student with me, went off, got a job at Conservation International, and came back and told me this story. And it's a story about this guy, Oliver Zampro, who was a graduate student in Germany, and he was supposed to be working on stick insect systematics. And instead of doing that, he went into the museum he was working in and went through the messy collections drawers. You know, we all think about these collections drawers, but every museum has got this. No, this is not my drawer. It's not dissimilar from my drawer. But so he goes into this drawer and he finds this beast, which is an insect in amber. And he looks at it and he thinks this is a new order of insects, and he runs up to his advisor and he tells him, I think I found a new order of insects, and his advisor says, why aren't you writing your thesis? And they go back and forth, and finally he convinces his advisor to write other people and ask them if they've seen this, and miraculously in another collection there's another one of these things, and it's on a pin, which meant somebody collected it alive, and nobody seems to know what it is. And so maybe it's this cool new order of life. And the good news is those specimens on the pin are from this Brandenburg Massif in Namibia, which is out in the middle of nowhere. Good news because it could still be there. Bad news because Oliver's a grad student and he has like $800 for the next 4 years of his life. And so this is when my friend Peter comes in, because Peter bankrolls this project to go and look for this insect. And so they go out with these cards and they say, you know, excuse me, have you seen my little bug? And people say, no, who are you? The whole thing is weird. But then they helicopter in to the top of the massif carrying all kinds of gear, and then they're going to walk out. And this is so exciting to everybody that a whole bunch of entomologists have come with on this trip. And it ends up being important that they're not all in terribly good physical condition. But they go and they look, and they see this, and they see this, and they see this. And they start to question the mission. They start to question their careers. A couple of them are doubting their relationships. And then it looks like this. And they're looking for an insect this big, right, which might or might not still exist. And then they find them. And they're these beautiful insects. And then they find a second species. And it's just this magical thing. And now they've got to hike out and show this to the world. But they've got all this gear. And so they start hiking out, and they can't carry everything. And so they start leaving cameras, and then food, and then clothes. And it's not totally true, but I kind of picture them walking out in their underpants, holding these insects on their head. And so they come back to the world. And because Oliver's a grad student and doesn't always make good life choices at this point in his career, he calls this the Gladiator Insect because he was watching the Gladiator when he first saw it. And he publishes the report about it, and because he's an entomologist, he publishes it on the basis of how unique its genitalia is. And what Oliver thinks at this point is that he's going to do this report and that the world is going to look at this and say, oh my gosh, Oliver, the way in which you've discovered this new genitalia, it really changes everything, which is kind of what happens. This is in the New York Times, it's on Time Magazine, it's a big discovery. But then what happens that's different is other people look in their insect drawers and find that they too have this new insect order. And in fact, in South Africa, it turns out to be relatively common, including in abandoned lots behind schools, which is to say that if kids went behind their schools and looked for insects and collected them, that they could have found what ends up being a new order or suborder of insects, depending on who you believe. And so I started to write this story up, and this would become the kernel of every living thing, except like all things around which books build and seem like the epitome of the book, it eventually got cut from the book totally. And then it was sort of the end of the story, except that Peter then went back to South Africa. Peter hates cities. He's sort of rank order. He likes orthopterans, other insects, other animals, people. And so Peter's in Cape Town. He really wants to get out of Cape Town. He hitches a ride with a trucker. Trucker stops at a truck stop. Peter looks for insects. In a bush at the truck stop, Peter finds 4 new species of katydids and another species of this gladiator insect. Which is to say that not only are these things everywhere, these kinds of discoveries, but you can find them at truck stops. To me, it was amazing. And so I really thought about what else was left to discover, what are we missing all around us. Every living thing was a testament to that idea. And then I started thinking about how was it that we could be missing things like this. And I heard this phrase the other day, and I really liked it, and I think it has a lot to do with the idea of the arrogance of now. We assume we mostly know what is knowable, and that what's left for us to discover is little stuff. And so as a grad student, I felt strongly that what was left for me was little stuff. And Peter's story really kindled this idea that there was big stuff left. And so one implication of this is that grad students should never be doing boring things. And so if we think about how much is left to study, which is what that book framed, this is an estimate that Linnaeus had of how many species there might be on Earth. This is the one estimate about how many are named today. This is a conservative estimate of metazoans living terrestrially, the most conservative estimate. Terry Irwin every so often suggests 100 million. I think we can't rule out 200 million. And what I like about this is not that I believe this 200 million circle, it's that we know so little that nobody can rule it out. And so just as a measure of this, we've been working on houses across North America. We just have people swab their house, and then we look at the DNA on the swab. In doing that, we've now found more kinds of archaeans inside houses than there were known in 1990. These beautiful single-celled organisms. Bacteria are ferociously diverse, not surprising, but this is relative to mammal and bird species on Earth. But the real kicker for me and the reminder of the arrogance of now was that when we did this, the number of fungal species we were finding in houses, this line right here, is more than the number of named fungal species on Earth, or in North America. And so all of this is a reminder of this, there's still big discoveries everywhere around us. But when I talk about this, often if I talk about it to the public, there's this question of, great, dude, but who cares? And so I've, later in my career, started to think more and more about this question of who cares and why does it matter that we don't know any of these things, and why does it matter that these big discoveries are still possible? And what I'm going to argue today is that we miss nearly everything, that seeing often requires assuming nobody else knows, and that the value to humanity often requires linking discovery, natural history, ecoevolutionary theory, and then importantly, human needs. But that in most institutions, those are all considered by different people who assume somebody else is doing the linking. And it seems like there's a lot of special work happening here across disciplines that's really rare, but generally speaking, those fields are done in isolation. And so I'm going to walk through some stories from the Hart book and then from other work I've done that I think are emblematic of this narrative. One is the story of this guy, Akira Endo. He grows up in Japan. He's born before the war, but grows up in his teenage years after the war. And he grows up in a time in which collecting fungi is just not just interesting and good for fun food, but also important to feeding his family. And so he grows up learning about good fungi to eat, bad fungi to eat, and then dangerous fungi. And he decides as he becomes a young scholar that he's really interested in fungi, and for reasons that are unclear, decides that if he can think about the ways in which fungi and bacteria have been at war, that maybe he can use the knowledge of those ancient wars between fungi and bacteria to do useful things. Maybe he can think about ways to solve diverse societal problems. And so he's thinking in some extent about Fleming, he's thinking about discovery of antibiotics, but he's also thinking about what are the diverse uses of that insight. And so he goes to the US, and in the US he notices that lots of people are suffering heart problems, and that those heart problems seem to be linked to cholesterol. And so he goes back to Japan and he tells his boss, what if I dedicate myself to finding a drug that could lower cholesterol by looking to fungi to see if any fungi have tried to interrupt the cholesterol-like pathway in bacteria in order to compete with them. His boss says, great, you're doing great, you're a great young scientist, why don't you try for a little bit? And so he tries for a little bit, and after 3,000 fungal screens, he finds this compound citrinin that works in rats, and he thinks he's finally done it. This has taken 3 years. His bosses are a little bit reluctant about still paying him, but it gives the rats kidney stones, and so his boss tells him, time to quit, you got to stop doing this work, you're done. And he says, no, this is really important, I'm really committed to this. And so he checks 6,000 strains. In 1973, compactin, which came out of one of those strains, works in Petri dishes. But he tried it in rats, and it didn't work in rats. This is now 6 years in. His boss has been paying him for 6 years with nothing good coming out of it. His boss tells him, it's really time to quit. You're done. I actually pay you. I can actually tell you to quit. But then he's walking home, and he finds a friend who has chickens, and he says, hey, can I borrow your chickens? It's not really a borrow because he's not getting them back. And so he tries this compound from the fungus in chickens, and it works to lower the cholesterol in chickens. And this is the compound that will become the precursor to statins and lead to the blossoming of the statin industry. It's really an industry now, which I think is a big part of the reduction in cardiac heart disease. And the fact that we got there in the first place related to Akira Endo's attempts to think like a fungus, to think about how fungi fought with bacteria, and to use historic collections that traditional fungus biologists had collected across Japan. Another example, this is the first attempt at a heart transplant. Christian Barner did this. This was a heroic attempt. Louis Washkashki lived. And so Barner was on magazines around the world. He was a hero. He slept with movie stars. His wife didn't like that. And this is part of the story we typically tell, but the other part that gets told less often is that Washkashki died in 18 days. And so realistically, that was a failure, and it was a failure that led to many other failures if we measure it from the patient standpoint, because the next years would be the race to be the first to do a heart transplant in the US, and then in the southern US, and then in Florida. And so many, many heart transplants happened, and the reconstructions suggest that, on average, they weren't extending the life of the recipient. But they were showing what we could do. What really happens is that cyclosporine is discovered, and cyclosporine is a drug that suppresses the immune system, and once we could suppress the immune system, and once we'd done a better job of figuring out how to do the transplants in the first place, Then they took off, capped out by the supply of hearts and to the use of seatbelts. But this then becomes the story, and it's traditionally told as a cool way in which industry and chemistry looks at soil fungi and changes the future of heart transplants. This was the secret, the cyclosporine. It was found in a fungus in the soil, and that was it. But then Kathy Hodge at Cornell University one weekend was looking at fungus collections, and there was a collection that was made by students, and she thought, well, what would be a fun way to spend my weekend? Why don't I try to grow up this fungus that was collected in a beetle larvae found in cow poop? Because as biologists, that's the kind of thing we do on the weekends. And so she did it, and she grew it up, and turned out that this fungus that you see here, which is a Cordyceps fungus, was actually the same fungus that produces cyclosporine. And so the interpretation, and so we know that Cordyceps fungi take over the brains and immune systems of insects and make them do their bidding. In this case, this beetle crawls into feces where the fungus grows really well. And the interpretation is what's happening with this cyclosporine producing fungus is it's taking over ancient parts of the immune system of insects that are sufficiently shared with humans that's why it works. There's been very little follow-up on this work. Another example, we know that we're super similar to chimps and that this is a beautiful thing. We assume that given the same circumstances, we and chimps die in similar ways. These are the beautiful coronary arteries of a human heart. And so for most of the history of the study of hearts, to the extent anybody thought about chimp hearts, the assumption was that chimp hearts can fail like ours fail. And in fact, there are lots of records of chimps dying of heart attacks. And so everybody just assumed that when they died of heart attacks, I mean, what more human moment is that could you have than this, right? Like this is our kind of sorrow, must be our kind of heart suffering right here. But nobody really looked. And so Nissi Varkey decided that she would go back through museum collections and look at parafilm hearts of chimpanzees who had suffered and not suffered from heart attacks. And long story short, what she found is that the chimps who had suffered from heart attacks in almost every case had fibrosis of the heart, where the muscles form these cross-links. This is the kind of heart attack that humans have not been shown to suffer from. There were no examples of heart attacks due to atherosclerosis. And so it appears that there are now two mysteries. One is why humans are suffering from a kind of heart attack that chimps aren't, and the other is what's causing this. We probably won't resolve this mystery now, I would guess. The first answer was that the cholesterol levels were different in humans and chimps, and it's pretty clear that that's not the case. If an infant chimp came into the hospital and the doctor wasn't paying attention, the infant chimp would be given statins, almost certainly. They have high bad cholesterol. And so that's a mystery that's not been resolved, but it's been reframed by evolutionary thinking. Another aspect of this evolutionary or sort of ecological thinking was the question that if the chimps and humans are not suffering from heart attacks in the same way, when did we start to suffer from heart attacks in the way that we do? This required museum work, and it turns out that you can do a CAT scan on a mummy, and so you can look for atherosclerosis. The assumption in the literature for the most part, although there's an old literature that doesn't get cited on mummies, but the most part the assumption was that these mummies would not have atherosclerosis or be very minimal. It turns out that in fact the Egyptian mummies have relatively similar levels of atherosclerosis to a dude in New Jersey watching TV and eating Cheetos. The first response to this was to say, well, these were mostly the royal Egyptians, and so they were eating like us, they were living like us, they were getting carried around in the way that I got brought here in a golf cart today. And so then somebody went and thought, well, let's check. We actually have lots of mummies from around the world. And this is a nice study showing atherosclerosis across different groups of humans through mummy work, showing that it's prevalent in very, very different cultures with very, very different diets. And so suggesting that there are these key transitions between their shared ancestors with chimps and our human story that need to be resolved and are only going to be resolved with these comparative kinds of studies. Another example, once I started thinking about this, I realized I'd written about more of these than I knew. Going back to Toxoplasma gondii, which probably folks in this room mostly know about, but 10 years ago we only ever heard about in the context of kitty litter, right? That if you're pregnant, you don't mess around with kitty litter. I, in fact, don't recommend anybody should mess around with kitty litter. But And the reason is because the fetus can get infected with this parasite, which is really bad news. And then there was this separate thing, right? And so this was medical, this was funded. This was ecological, this was not funded. People studied this, and ecologists knew about this story, but nobody else did. And this was the story that the oocysts have to get into mice and rats, but they can only have sex in a cat. And so they have to have weird ways to get into a cat. And there was a really nice body of work that suggested that Toxoplasma gondii makes mice and rats less scared, less vigilant of predators, attracted to the smell of cat pee, and more likely to be eaten by a cat. And part of how they do this is they produce the precursors to dopamine in the brain. This was beautiful work. It was poorly funded work. It was work being done by basic biologists that the world didn't know about, but basic biologists knew about. And this is the kind of thing that you would hear on NPR of like, why are we funding this work? And one of the reasons that clearly we shouldn't fund this is because it doesn't relate to humans. Because although we get infected this way, it's not affecting our behavior. Although there is this weird thing that if we eat meat that's not fully cooked, we can actually get the other form of the parasite. And so at least in theory, if you were a circus performer, you could get eaten by a cat, and then the parasite could have sex in the cat. But this was a weird theoretical possibility until it became clear that there does seem to be this effect of Toxoplasma gondii on behavior in humans. All the early studies were correlational. Now there are some new studies that build on this, but early on it was suggested that people who are infected with the parasite have increased activity, decreased reaction times, altered personality profiles, 2.5 times more likely to be in a car accident. And then, and this is the study that really is fascinating, and through time studies of enrolled military people showed that the single best predictor of the onset of schizophrenia is infection by Toxoplasma gondii, which is not to suggest it's a sufficient causal agent, but that it's part of the story. And so suddenly this becomes an important human story, but the only reason we know about it is because of that basic work. And then coupling that with humans, turns out it's potentially explanatory in other ways. 80% of the French have been infected by Toxoplasma gondii, so it could explain a people. And probably half of you in this room have been infected. And so as I started to think about these stories, one of the things I started to think is, could we think about how to do this systematically? How to combine evolutionary and ecological work with insights about societal needs, and to bridge these things more often. Because when we write to NSF or when we write our papers, there's often that first paragraph that says the reason this is valuable, and then that's where we then write something we don't believe but we think other people want to hear. And we train graduate students to do that, right? I want to ask for a show of hands how many people have written a paragraph in the intro to their paper that they don't believe but they think they need to have there. And so we started thinking, how could we do this systematically? And so we decided to look to our evolutionary history, look to other societies and species that have met our challenges, and look in light of species we encounter every day, but to do this work in partnership with people who are thinking about societal challenges. And so I'm going to walk through some case examples here, and then I'm around for the rest of the day if people want to talk more about these things. And so first, looking to our evolutionary history, we started a project on belly button biodiversity, which was perhaps the silliest thing I've ever been involved in professionally. And it was an effort to get people thinking about what lives on their skin, to think about this complex ecosystem, the extent to which it's there, and to which they're probably making dumb decisions about it. And so we did all kinds of things during this project. We engaged artists to help us engage the public. This is Joanna Rissu. This is Joanna Rissu's microbes on her head, right? How, if we were to smell Joanna's head, this is the version of her that we would perceive. But then the question about this work, as we started measuring people's microbes, was who cares about this stuff? And for the most part, we were happy not to answer the who cares question because we wanted to understand why one person has these microbes, why another person has the other microbes, what ecological rules manifest in determining which species you have versus somebody else. We loved the basic stuff. That's what we thought was important. We compared these microbes to tropical forest systems. But the truth was there were some interesting who cares parts to this. And one is that the first thing that happens when a pathogen encounters your skin is it encounters the microbes on your skin. And so there's an important pathogen piece. Another is that when a mosquito gets close to you, it decides whether or not to bite you on the basis of volatiles being produced by you, but also to a greater extent your microbes. And then another is that these microbes on your skin, they're in a constant interplay with your immune system, and so you can get interesting dysfunctions. And so we started to think about, well, could we look at the evolutionary history of these microbes and then talk to people who do application and see if we could do our basic evolutionary biology, but also think about application. And so we just started off by looking at what are the skin microbes in the armpits, in this case, of different primates. So this was a super simple study, and I'll just say some basic things about it. The macaques and baboons were really, really diverse. Gorillas and chimps and humans less diverse. And we're really dominated by what's Staphylococcus, but there's this Corynebacterium that's really common in our armpits across the apes. And so this was all new because it turned out nobody had really looked at skin microbes of primates. And then it overlaid with this other interesting thing, which is if you look at humans, this is the average story. But if we look from person to person, there was tons of variation that was unexplained. Whereas we didn't see that in chimps, gorillas, or the monkeys. And it turned out that this was actually, to a large extent, explained by deodorant and antiperspirant use. And so we did this nice experiment. This was this foul experiment, really, where we had people wear or not wear deodorant and antiperspirant, and then we had an experimental treatment with antiperspirant. And short story that, if we look down here, this is Corynebacterium. This is this thing that's more common in apes. It's really common in people who use no antiperspirant, and those who apply antiperspirant, it disappears. What appears in its stead is Staphylococcus. This is interesting because we're starting to show that mosquitoes don't like this. They love them some Staphylococcus, which is now interesting in a historical context. And so we're now working with applied biologists who work on probiotics and mosquito assays to think about what can we learn about these microbes that might allow us to think about probiotic clothing or mosquito nets that deters mosquitoes. So we can talk more about this, but this is just something I never would have done before and I could never have done on my own. But by working with people who are thinking about an applied need, we can do together. These are the proboscises of these mosquitoes poking through this mesh. Meredith Spence, who's deleting a lot of that work. A cool little aside from yesterday, really, is that we can explain part of the variation from person to person as a function of antiperspirant use, but there was still some unexplained variation. And we've now looked at people that differ in the ABC11 gene, which is associated with dry versus wet earwax, the single nucleotide substitution. And we can show that both in armpits and ears, that whether you're homozygous for dry, homozygous for wet, or heterozygous explains much of the variation from person to person in which microbes you have in your armpits and ears. This gene has been under strong selection, and so it's a really interesting one to think about. Explains a lot of what's going on with diversity in the skin. And so this is another fun part that we're working on. Another basic study that we started doing that had no applied value, we just thought it was cool. These are face mites. We've now found that everybody has them, and so they're on all your heads. They eat, they crawl out in your face and mate, they fill up with feces and have no anus, and so eventually just disintegrate. No biologists kind of like that, other people don't. And they have tons of structure, which I'd bet for years, and we never did this work, and finally we did it, and there's lots of geographic structure. There are really old splits in this lineage. There are 2 species known from humans that in 1970 were thought to be one species, and then people said, oh, they're kind of different, must be close relatives. It now looks as though they're 120 million years divergent and just dealing with these similar circumstances. So it's a really cool radiation. We think all mammals have them. Only 27 mammals have ever been sampled for them. And we're finding all kinds of cool basic biology just because we like these mites. And we engage the public a lot about these mites, and so we thought we need really good art. And so we went to the Smithsonian to get good SEM images of these mites to show kids. And so when we did that, we saw that they have cute toes. We saw that they got the no anus. And then we went and looked at their mouth parts, which have been known for 100 years to chew at your cells and then imbibe what they chew. And we found that they have no chewing mouth parts, which we only found because we wanted to do art on their faces. And so now we have to figure out what they're eating because they're not eating what anybody thought. Who cares? I didn't care who thought who cares, but now we're thinking about this. Well, these mites are implicated in rosacea, but they're clearly not causing it because everybody has the mites. And so one possibility is that people with rosacea have a different mite to which they're generating an immune response. And so we're working on this now. Because nobody's sampled dogs or chimps or gorillas, we have to do a lot of work to get to where we can know that. But the other thing we've started to think is that what's interesting is that if people are generating an immune response sometimes to these mites, that most of the time they're not. And so the mites are almost certainly producing immunosuppressants. And so we're now working with a group that works in immunosuppressants to see if we can find new immunosuppressants in these mites. We are also looking to other societies and species that have met our challenges, fungi and ants, carrion beetles. We're looking for new antibiotics in light of these beautiful carrion beetles, because you bring these carrion beetles into the lab and you give them their dead meat, and it doesn't smell perfect, but it doesn't smell strong. And so they're almost certainly both culturing interesting microbes and producing antimicrobials. And so we're teaming up with people who do antibiotic work to see what we can find in light of that system. We're also looking to species we encounter every day but overlook. And so we're doing that a lot in homes. I'm really interested in this transition, between homes like this that we all lived in 12,000 years ago and homes like this, and what happens to the species we live with in that transition. And so we've now looked in homes, and we've asked people to look in homes, and we've found things like this great camel cricket. Camel crickets have been known to be in homes for a long time, but we just asked people, where do you have them? This is a map of where people said they had them. This is where they don't. This was clearly wrong, because we know from the systematic revisions. And so we asked people to send pictures. They sent pictures, and lo and behold, the camel cricket in people's basements is this invasive Japanese species that we essentially didn't know had moved into homes. We then asked, who cares? In fact, we didn't ask. People asked us a lot on this one. And so we thought, well, what good would this cricket be? And so we teamed up with people who work on biofuels to break down lignin. And we thought, can we look for bacteria that break down lignin in these camel crickets? Because that would be interesting, especially in light of paper pulp. And long story short, we've now found 5 species of bacteria that break down lignin in the guts of these camel crickets, which adds by 40% the number of species on Earth of bacteria that can break down lignin. And so it's interesting, who cares? We've now gone on to sample lots of other things in homes. We've now found something like 2,000 insect species in homes. We found this great spider which spits silk on its prey. It's not known outside. World's smallest cricket. It's in my house. I don't know why. And then we've also extended to we now sample using genetics approaches, and so this is just microbes in homes. Quickly, this is just a figure of, showing that here we get things from the closer together 2 points are, the more similar they are. These are things associated with food. This is somebody with sausage on his TV. This is outside associated things. This is the reality that we can't tell toilet seats and pillowcases apart. We can tell their gender because there are lots of vaginal microbes on women's pillowcases, but we can't tell the two apart. And so we now have this big study to sample. We've now sampled all these houses. The orange dots are places where we have full genomes of the humans in the houses, and we're now working to pull these whole stories together to figure out, do the genes of the humans in the homes affect which species are living with them. We can also now do maps of the 150,000 species we've found in homes. And we can think about cool things like lactobacillus bacteria we use in sourdough. We're essentially measuring the terroir of your house. We're measuring what's falling into your sourdough when you make it. Again, the who cares. We didn't know, but then I got a call from the CIA at 11:30 at night one night when I was doing laundry, and they said, can you use these data to find out where a sample is from? Turns out we can. We can tell where a dust sample is from within 100 kilometers based on the fungal species in the sample. What this has led to is for us to think about other systems and what else we can predict. We're actually now using the ant DNA from these dust swabs to try and see if we can do a better job, and we can do it globally. And so to come back to this talk's thesis, I really firmly believe that we still miss mostly everything. And that seeing often, I hope these examples show, requires assuming nobody else knows. But I think it also requires teaming up with people. I can't do any of the work that we're doing now without somebody who knows the application. And in most cases, it just required meeting somebody over beers and talking. And sometimes I meet mean people and I don't work with them. But I think there's huge opportunity. If I were a really good speaker, this is where I would say that this gladiator insect has now cured cancer. I'm not that good a speaker. But I think what we should remember in thinking about things like this new order or suborder is that we can only start to make these connections, start to think about these values once we know these species, once we've characterized them, once we've done the great natural history combined with genomics, like Jürgen gave me a look like the work that Jürgen's doing, that pulling things in from outdoors and characterizing them enough so that we can borrow insights from them. And the truth is most stuff is thousands of years from making it into the category of being something we can gather insights from. And so in conclusion, I think that ecological and evolutionary insights make discoveries more likely, big practical discoveries, but those insights can only be applied to species we know well enough to make predictions about. Most species are not yet in that category, and I really think that most species won't be in that category for 1,000 years unless we change very dramatically what we do. And so I'll close there. I think there's a 1 o'clock class, and so maybe a couple of questions, and I can run out the door while talking. But thank you very much for having me here. It's really a very special place you have here. Congratulations.
All right, so we have plenty of time for questions. The floor is open.
So I really enjoyed the talk, and I could not agree with your thesis more. My question is, we can estimate that one finding out of 100 can have huge translational implications for humans or some aspect of the industry. So how do you communicate these concepts to funding agencies or venture capitalists or other people? Because everybody wants to be the one that's investing in the thing that's going to be the hit. But in order to get those hits, you have to invest very, very broadly.
So I think we don't actually try. So I think that what we've built, the diverse collaborations here as an exception, are institutions where we have a group of people who are pursuing very deeply these basic things, and these applied people over here, and then we assume there's a group in the middle. And that for the most part, at most institutions, there aren't those people. And so, I mean, history will say whether what we're doing now, any of it's useful, but I think looking at other examples, they very often involve pulling people from two of these fields across to have conversations about what do needs look like and what do you know. And just an interesting example, I went to Elon College, which is a liberal arts college after my own heart where I went, and a student asked me, we have a paper on stomach pH, and she said, why didn't you include bearded vultures? I go, oh, I don't know. And so I looked at bearded vultures, and it turns out bearded vultures have this stomach pH of 0.7. I then went and talked to an industrial microbiologist who's working in these really acidic contexts, and she's looking for microbes that can grow in that condition, and her conclusion is that there's no place good to look. And so all it would have taken was an undergrad meeting with that person, and I don't know if that's going to work out, right? But on average, we very rarely have those conversations. And I think that, one of the hard things is sometimes we think that a single person should walk across that bridge. And I think that, especially for young scholars, that's a really hard place, right? Because you're on a tightrope, and underneath the tightrope is your mental health. But I think if we can build ways for groups of people that go across those things, I think it would be really useful. And I think just in the basic biology in this room, there are tons of things that could be extended to application that the basic biologists won't know the need, and the people who have the need won't know who to talk to. And so somehow we have to get better at that. And I think we've on average, I don't know, does that answer your question?
Yeah. How do we get better at that? How do we get investment in that? How do we get people buying on that? How do we, showcase, how, people have to pick up your book and read it to understand some of these things, and they have to read other people's, how do you get broad buy-in to building these bridges? ASU is doing it, but we need everybody to be doing it in greater scales, I think.
I think grad students are a great place to start. Like graduate programs where you enforce is wrong, where you provide incentives. Or maybe you enforce, I don't know. You enforce, but you provide incentives for graduate students to span disciplines. And so I think biomimicry here, is a neat example of that, where you have people coming across groups to work together, and there's a supportive framework for that to happen. And they don't have to believe that they're going to make the next great discovery tomorrow. They just have to believe it's normal to talk across that divide. I always think it's hard to get a group of senior people together and convince them to do something totally different, right? It's harder to enforce. But I don't know, I think we've discovered some really wonderful basic things that should be of value to society at this point that aren't. Like milk's a super interesting case example, right? Like right now, most of the bacteria we use in yogurt come from very few sources. And if you look globally across milk from different mammals, I guarantee there are more interesting ones that are doing things that would be more useful to yogurt. And it wouldn't be super hard to try, but you need somebody who can industrially think about what would you need to do to bring that to scale. And yeah. Other questions? Yep. Tell me your name when you ask the question, too.
This one, so I should be fine. Well, maybe a little overly personal and specific, but about your Demodex mites. So I thought it was interesting that you found all this stratification, like the population structure, from different continents. I have rosacea. I spent my childhood living in Japan, so I can't help but wonder, do you see any kind of this? You see extra prevalence of it in people who have done a lot of traveling, or you have no?
So we don't know now. The thing we're doing next is to sample a bunch of people with rosacea to see if the mite lineage they have is different. Okay. That presupposes we've sampled enough the bigger tree of the mites to know what that would look like. And so we're in that at the moment with Michelle Troutwain at the Cal Academy. So right now, some of the mites we're finding on 2 people with rosacea, their mite lineage more closely resembles a dog lineage. But the tree is so poorly sampled that we can't tell if it's a jump from dogs onto humans or if we've just missed everything in between. And so it's early. They also have these intracellular symbionts that are doing weird things, and then gut symbionts. And so it's been suggested that their gut microbes are somehow different on some people relative to others. But it's super open. But I think it's clear that we need this. If we're going to make sense of it, the medical framework for it is going to miss the story. So I wish I had a better answer. Yeah, I'll send you a swab for sure. Yeah.
So I have this idea that a lot of the things you brought up are basically everyone should do basic research, and eventually down the line it will lead to things that help humans live longer. But I have this feeling like we could do more with basic research that helps people live better, even through just learning about different species. Do you think this is something that's do you think there's an increase in interest in just basic science for the story? And is there anything you're doing that is promoting it, or good ways to?
Yeah, so I think that's great. So I think that the intro to why we do basic science is mostly a lie, and that it's a lie that runs different ways. And so one of the ways it's a lie is that we try to do intermediate science that's not quite basic, but appears to maybe be applied. But if you look at If you look at what has a big impact about how people learn about nature, it seems as though classic stories of interesting natural history are disproportionately important. And so really unpacking beautiful classic stories of how a system works, I think has huge value. And it's a different talk, but I actually think as we communicate to the public, that the ability to unwrap a story and how it was studied, how did we learn this, I think becomes really important. And that in K-12, we're doing a really poor job of talking about the process of science. And so if we can think about how do we build into talking about these classic examples, the classic story of how we got to that example, and also what we might still be missing, I think that's super important. And if you think about even as grad students, young scholars, that how you frame what you talk about and know. You're often doing it on these pillars of classic examples. And so I think they're really huge. And I think one of the things it means is if you take one of these things that's in the textbooks, going back and revisiting what we're still missing about it, like that should be extra well-funded. And so one I would love to do is to go back to the original studies of spontaneous generation and to work through, well, what's actually going on in terms of the microbes and the insects and the biology, and what do we know or not know about that example. And so we can then use that additional science to help tell the story about that. Yes, so amen to that, to your question and point. Yeah. Did that answer? That didn't look like a satisfied look.
That's a really great speech.
Totally irrelevant to the question.
I guess I just wanted validation, and also I'm wondering, are there any more, I guess even job openings or interest in that? Are more maybe museums looking for people who will talk about science versus just talk about how science can make you a healthier person and live to 100?
Yeah, so I'm delighted to talk more about that after, but I think there's a growth of hires right now. That's really a who's hiring what question, but there's a growth of hires right now that are trying to bridge science and engaging the public. And so the Cal Academy right now, for example, is hiring 4 to 6 people who will do important, interesting science, quotes, and engage the public. And so I think it's something that around the world museums are actively doing battle with. How do we best do that? And the Cal Academy is an interesting example because they have a huge engage the public team, 300 people, and then they have 100 scientists. But they're really very separate. And so I think one of the questions they're asking is how do we get the greatest stuff that's coming out of our scientists or scientists in general and have that be part of how we're engaging? And what does that look like? So I actually think for the kind of thing that you are talking about doing, it's an exciting time, but it's a time when people aren't sure what the answer looks like. Yeah, that looks more satisfying. Yeah. You're good?
More comment to the NSF. They should fund more discovery-based science. Man, I've talked to this driven science, and let's just kill. No, but really, I think that the more important question is the university and us as professors, right, having all these kind of trade-offs we have to deal with in terms of getting grant money, funding our grad students, and teaching, etc., and going through that process. It's really difficult to get the money funded to do these, right? I do a lot of collaborations and I love it. I think that's the most single important thing for young people to learn, really kind of talk to people and go to these and bridge across that. And I love that talk you gave here. But the other component, I think, is often things are rarely by chance, right? If you think of how you make this discovery, but then the next step is not falter, right, and just keep persisting.
Yeah.
Right, and say, okay, now that I discovered that, now I'm following a path for a couple of years, or I really want to find out the details.
So I feel like I have a bunch of things in my career where I failed to do that, and I can reflect on that failure. And so one that's, well, just to be honest, right? Just as one example that's really in my head right now, when I was a grad student, my wife and I did a paper on the medicinal uses of insects globally. She's an anthropologist, and so we went through this old database of ethnographies of the world. And we found lots of uses of social insects. Very often they're related to antimicrobial type uses. And there was this amazing use in Sulawesi of this ant that was kept in a bamboo cage and fed popcorn, that when somebody had an infection, its feces would be spread on the wound. And then there was this separate sort of cultural context, which is that if you didn't feed the ant, all of society would die. And you also had to tell the ant if someone died. That was another. And so as a grad student, it was clear that was potentially a pretty interesting thing to follow up on. But I thought, well, somebody else will do that. And nobody did. And so, just now, I refound this thing, which I couldn't find. And so I then thought, well, who can possibly help me figure out what this ant might be? Because it's a red ant in Sulawesi. Oh. And so I found the one linguist who's now 94, who's done a lot of work on this language, and he wrote back this 2-page thing that this is the weaver ant. This is very clearly, I've seen it used. This is the ant that's called Leah. Its different life history stages have different names, and this is a place where they do popcorn, and they might have fed some of it too. And so it was all this context that was available right now. If I would have asked him in 2 years, or I don't know, maybe he'll live to be 140, but it's a narrow window, and I wish I would have followed up on it 10 years ago. And now it turns out there's some nice work on weaver ants showing microbial inhibition from gastric secretions that makes sense in the context of a lot of the classic work, the classic stories on weaver ants.
But the problem is, what do they do with the popcorn?
Yeah.
I certainly don't eat it.
No, yeah. So that's the last mystery in this. I thought nobody was going to call me on the popcorn.
I just wanted to say, the context of the discussion before, since I worked in 2 cultures in the United States, and I know both very well, the funding policy, and I have to remind you, I was here active at Harvard when Broxmeyer was alive and created this otherwise great senator, this Golden Fleece Award for the most silly research project NSF is funding, and his team just looked at titles, and those titles which looked funny got the Golden Fleece Award. And I always said, as much as I like the United States and the academic world, the funding process is in trouble, because it is not entirely in the hand of scientists. It is interfered with by ignorant politicians, congressmen, senators, who have no idea. We have currently again one who is trying to get a profile in doing that. The difference to the German funding system is the political decision is how much money do we politicians want to spend for research and education. And then once this decision is made, the money is given to scientists. And then the German Science Foundation is entirely run, and all the decisions are entirely done by scientists. And they have to just, if they make a wrong decision, this is not their thing. I think this is a better way to do it. On the other hand, what I often want to remind the Americans is, if we only fund so-called society-relevant research, we will run into a dead-end street, as all the Eastern countries did during the Cold War. That this killed any good science in the US. And I bring always as an example Watson and Crick. They were not interested to cure cancer, they wanted to understand the alphabet of life. And look what came them. In retrospect, think, I know, Asen, that this is in connection with what do you tell the young students. I remember very well when I started my graduate work. I wanted to do something. I was interested in behavior and in ants very early on. But I wanted to do something important for society. So I decided to become a forest zoologist. And my first papers are published in forest zoology. And, but when you do this, you encounter ants all the time. And then I was drawn back, and I discovered we can only save this once we really understand the behavior. And so this brought me back to behavior, and I joined the Lindauer group and really do behavioral physiology. I do not, as you all know, it started during the Broxmeyer time. We had always to write 2 summaries, and we still do, for 2 abstracts for NSF proposals. One for the Congress people and one for the scientists. And I agree with you, a great deal is lying we put in this, because it's easy to invent something, why it is good and good for application. But it's a very bad way to educate our young people. I am for it that we stimulate everyone who has a gift should go out and explain science to the public. You do it wonderfully. But we should be honest why we do this. We want to understand life on this planet. And you know, it can be studied at so many different levels.
So I feel like we need to be honest on both sides, that we need to be, when our urge is really to understand life on this planet, to be honest about it, at least to ourselves. And I think that the more people write something that they don't think is honest, the more it also changes what they believe. But then the other side is when there is the appearance of the potential for something translational, that we have to be honest about the fact that if we don't talk to somebody about it, there isn't another person to do it. And so in those cases, which are going to be the minority, but the minority in lots of science that's going on, that, I mean, to go back to the milk example, that there's not a person that's going to do it, right? Like if you don't do it.
There's a lot of us working across industry and health and basic lactation biology, but it's taken a lot of effort of people to reach across disciplines and go to each other's conferences and build those relationships. And it's taken a huge amount of investment from the dairy industry to facilitate a lot of that. So but one of the things that we run into is that, you know, on average it's 50 years from bench to bedside for many, many things. And part of that's because some things never make it, but a lot of people that are having to respond to shareholders or show returns on investments, they want a much quicker turnaround. And so there's a disconnect between basic scientists who like to move, I think, very judiciously, and clinicians that want to move after randomized controlled trials, and people that want to make profits that want it to have happened yesterday. And so there's an essential tension among all of these stakeholders that need to all buy in and subvert their own particular cultures in order to do it. And that, I think, is the big challenge, is getting people buy-in.
All right. With that, I think we'll wrap it up.